So, from the process of cellular respiration to this point, we have used oxygen as the last electron acceptor. And in that process, we have produced a great deal of ATP. Um, Brief review of that process one more time. Glycolysis to split the glucose, citric acid cycle to get rid of the carbons and to charge up our electron carriers, electron transport chain to produce a significant electrochemical gradient right across the inner mitochondrial membrane, and then using the proton motive force of that ion flow to turn the ATP synthase. Makes a great deal of ATP. But remember, the last electron acceptor in that electrochemical, the last electron acceptor in the electron transport chain is oxygen. What if there isn't enough oxygen? That's where life can still exist. If there's no oxygen present, okay, so with no strongly electronegative oxygen there to be that last electron acceptor, oxidative phosphorylation stops. Substrate level phosphorylation can continue because that didn't require oxygen to begin with. But the oxidative phosphorylation, where we got all of that beautiful ATP, the 28 at the end of the chemiosmosis, that stops. So, what can be done with just that little bit of ATP? So, the production of ATP with no oxygen is in two forms, either anaerobic respiration, which is a shortened electron transport chain. The final electron acceptor there is not oxygen. It doesn't produce very much energy at all. And either that or fermentation. Okay? Fermentation is basically a continued recycling of glycolysis. Glycolysis is a net energy gain. Glycolysis is exergonic. So if we're looking at this, we can see that producing glucose, splitting that glucose down to our 2 molecules of pyruvate does eventually get us 2 molecules of ATP. So we can use that to power a cell that's, well, either not very active or that cell's going to be prokaryotic, tiny, and doesn't need much energy to begin with. Okay. There are 2 forms of this fermentation. The first is called alcoholic fermentation, okay, which we're seeing here. It's alcoholic fermentation because of what it produces at the end of the process, not because it can't hold down a job. Okay, so glucose is going to be broken down into 2 pyruvate molecules through a couple steps. An intermediate step here, we're going to produce some ethanol. Okay, notice the recycling here of the NAD+. So our electron carrier is going to recycle, and so we have a very small cyclical process here that produces a tiny amount of ATP from our glucose. But notice the ethanol is left over. It's a large organic molecule byproduct that then has to be dealt with. Either it has to be metabolized, or if this is yeast, then that ethanol is just in their environment. Okay? And we use that. We use these kinds of yeast to produce wines, spirits, beers. We use those kinds of yeast to produce extra carbon dioxide for things like leavening bread. Okay? The second kind is referred to as lactic acid fermentation. Okay, and this is a one-step process. Okay, same kind of NAD recycling, but instead of moving from glucose to the alcohol, now we're moving from glucose to lactate. This is much more typical of a eukaryotic cell. This is what happens in our bodies when that oxygen depletion in a high stressful or high strenuous situation. This is what's going to start to happen. And notice we went from making 28 ATP through chemiosmosis to just a couple ATP in this lactic acid fermentation. You can't power a human body for very long with that amount of energy.